had to, let's see, it was either question 5 or question 1, depending which version that you had. Michaelis-Menten describes the velocity of a reaction at equilibrium. Remember, Michaelis-Menten equation, when what you're graphing is initial velocity versus the substrate concentration. So it's not the velocity after you reach equilibrium, it's the velocity before you get to equilibrium. And so A is not correct, D is not correct, and so that leaves B and C. Now, I guess I'd mentioned kind of in passing about the steady state assumption that part of deriving the equation is actually in its steady state. So I didn't. So what I ended up doing on that one, if you answered either B or C, I gave you credit on that one because we didn't really get into the fact that in that equation the change in concentration of the ES over time equals 0. That's one of the assumptions that's used to actually drive the Michaelis-Menten equation, but we didn't get into that out in big time. So I did B and C. Now the other one that I didn't change, but I did want to pull out because it represents an important concept. It's the one about if you know delta G naught prime, what don't you know?
Number 9.
Yours, it's number 9?
Mm-hmm.
Yeah, there we go. So if the value of delta G naught prime for given reaction, one may determine all the following except the. So you do know the direction of spontaneous change in standard conditions, because that's basically if it's negative, it's spontaneous. If it's positive, it's not spontaneous. You do know the position of equilibrium, because you can use the equilibrium constant equals e to the minus delta G0 over RT. And the stability of the products is related to the reactants under standard conditions. That's what it's telling you, is that the relative energies, and the lower the energy, the more stable the product. So that's what it's telling you. What the thermodynamics doesn't tell you is anything about the rate of the reaction. So again, this is something that a lot of the biochemical reactions that go on the cell are highly spontaneous, but they don't happen unless you have the enzyme in there, right? That you can have reactions that are highly spontaneous that go very slowly. Maybe one of the best examples is paper. The oxidation of the cellulose in paper to CO2 and water, right? If if you add a flame to that reaction goes pretty quickly. You'll burn the paper up, right? But under conditions here, it's kinetically stable. It's thermodynamically very favorable, but it's kinetically stable. So we can use paper to write on, and we don't have to worry about burning up. And somehow we add energy to the system by putting a flame in there. Then we have to worry about that. So that's kinetically stable in spite of the fact that it's thermodynamically favorable. And so most biochemical reactions are that way. Any other questions people might have? All right. Oh, the other thing is on the grade and Canvas. I did make one change for you folks who might follow it closely. So what it was doing, it was bringing over the homework points into the Canvas grade sheet. The problem with that is a homework point isn't the same thing as all our other points, right? So I can't remember exactly how many total points I gave the homework for the class. It'd be like 10 or 20% or something like that of the total class points, right? So like it had 120 points. Well, you're not really going to have 120 points in there. Once basically when we get to the homework, I'll look at your percentage score on your overall on your homework, and I'll multiply that by the number of points that I said in the syllabus the homework was going to be, and that's the number of points. So because typically the homework scores tend to be pretty high, that was basically overweighing the homework score. So I basically took that out of the calculation for now. And so the way it should be right now is your score not considering the homework scores. So, and I haven't figured out a way to actually make it make it, real time in terms of weighing it properly, so you just have to kind of keep that in consideration. Any other questions about that kind of stuff? All right, very good. Last name? Have fun at the planetarium show? It was kind of a nice break. There wasn't that much chemistry, biochemistry in there, but still, I think it was worth doing after an exam where you're kind of fried in. Kind of mind-blowing, all that acid astronomy stuff. All right, what are we doing today? Getting into the nitty-gritty of glycolysis, the metabolic fates of pyruvate, and then talking about the reverse of glycolysis, gluconeogenesis. That's my goal, is to try to get through that. I might not get quite there, but we'll give it a shot. My approach for trying to make up for the fact that we had the holiday, that I hadn't realized that we were going to be in there, is to kind of nibble away at it, basically. But I want to point out that I do want to make a change, though, because I don't think I can nibble away that quickly, is that this will change the midterm next week from Wednesday, which was originally scheduled, to Thursday, okay, to give us a little bit more time before that next midterm. And I adjusted dates on homeworks a little bit, so you can go and there's all the homeworks that are due. Don't forget the old double helix thing. I did get a couple turned in, so somebody's on it. Any other questions about this? Where we're at, what we're doing? All right. All right, so last time we had introduced glycolysis. Remember, put in the big picture, we're totally oxidizing glucose to CO2 and water. And we're going to couple that oxidation to the synthesis of ATP. And remember again, what oxidation is really meaning is we're taking carbon-carbon bonds and carbon-hydrogen bonds and converting them to carbon-oxygen bonds, which are inherently more stable than carbon-carbon bonds, carbon-hydrogen bonds. And we're going to capture that energy to make the phosphoanhydride bond of ATP. And then that universal energy currency is going to go around and drive a whole bunch of different processes, whether it be contraction of muscle or making the sodium-potassium pump go so that we can power our nervous system, or a whole host of different things that use ATP as an energy source. And so we talked about, again, how we have glycolysis, and that makes pyruvate, and then we go to the pyruvate dehydrogenase and the citric acid cycle. And those processes basically are the processes that will fully oxidize glucose to carbon dioxide, but we're going to capture that energy in a high energy electron carrier, mostly NADH. I'll give you the structure of that a little bit. And then the last section of it is the electron transport chain, And we're going to use that high energy electrons to drive the electron transport chain, which is going to allow us to make ATP in a process known as oxidative phosphorylation. So we're the beginning part of that process of glycolysis. We said there were 2 phases. What was the first phase? You remember? No. You
Use
remember,
energy.
Tiffany? What's that?
It's the use energy phase.
Yeah, so there's basically an energy investment phase and an energy recovery phase. And so we're going to go through the 10 reactions. We'll start with the 10 reactions, begin in the energy investment phase. But I kind of want to point out something before we get into that, is I'm going to take glucose, and I'm going to draw the Fisher projection of glucose just to make a point here. So the end of this first phase is the generation of 2 glyceraldehyde 3-phosphate molecules. So we're going to start with glucose, and we end up with 2 glyceraldehyde 3-phosphates. So we can think about, okay, what are some of the things that we have to do to this molecule to convert them into this molecule? What's one thing? Yeah, we're gonna have to break this, right? Now, it turns out that the chemistry that's involved with cleaving this bond requires a carbonyl at this position. So one of the things that we're going to have to do is we're going to have to move the carbonyl from this position down to that position. Anybody remember the name of the 6-carbon sugar that has the carbonyl at the 2 position rather than the 1 position?
Fructose.
Fructose, right? So we're going to have to isomerize it from a glucose structure to a fructose structure. Okay. What's one other thing that's different about the glyceraldehyde 3-phosphates relative to this?
It has that phosphate.
It has phosphates in there. We got to add, and since there's 2 of these, we have to add 2 phosphates. So one of the reasons I'm talking about this is because there's 10 reactions. You can memorize them brute force, but you can also, what helps on the memorization is if you kind of understand, okay, well, what are we doing here? And then you say, well, I know I need to add some phosphates, and I know I need to change the structure from a glucose structure to a fructose structure, and that just helps with the memorization. So let's go ahead and start with the first reaction, and the first reaction is going to add one of the phosphates on there. So this will be, actually let me like this is number 2. I did like an introduction talking about the 2 different phases, so this is sort of the reactions of glycolysis. And I'm just going to go ahead and put numbers because I like to number the reactions rather than letters. So I'm just going to start. 1 is the reaction catalyzed by hexokinase. And here we're going to take glucose. And I've used this reaction as an example in several things throughout this course, so hopefully it might look a little bit similar to you. And we're going to take a phosphate from ATP, and we're going to phosphorylate the 6th position. So this is glucose. And this is glucose 6. And I'm going to do this abbreviation just to help us get it through. I could write phosphate each time, but I'm just going to put a P with a circle, because a P with a circle represents a phosphate. Now we'll talk a little bit about the thermodynamics of the reaction and ultimately talk a little bit about this. But I'm just going to give you, and you don't have to memorize these numbers, but you can kind of memorize whether or not this reaction is highly favorable or not highly favorable. And so this, the delta G naught prime for this reaction is minus 18.4 kilojoules per mole. So, you know, what that does, if we have 1 molar ATP, 1 molar glucose, 1 molar glucose 6-phosphate, right, under standard conditions, this reaction is going to want to tend to move to the what? To the right, right? It's going to be favored. Oh, this relates back to one of those questions on the exam, though. But, you know, this compound here is going to go on. There's going to be subsequent steps, right? It's going to keep going. It's going to keep moving, right? And so we're going to reduce the concentration of this. What happens if we reduce the concentration of this from where it would be at equilibrium? What's going to happen to our delta G? It's going to make it more favorable. It's going to be more negative. That was a question on that final. So as we consume the product, and you know, mathematically, how do we show that? Delta G equals delta G equals delta G naught prime plus the log of the ratio of the products and reactants. So you can kind of go back and do the math, kind of show you that, okay, well, if we consume the products, that's going to shift this like this. So if we look at delta G, and remember delta G takes into account what the actual concentrations are of the reactants and products, and this is going to depend upon the cell and the conditions. But let's see, I think that these come from like rat muscle, something like that, rat cardiac muscle, if I remember right. Just as one example, the delta G is minus 27.2 kilojoules per mole. So not only is this reaction favorable, but under the conditions of the cell where the product is going to get consumed, it's really highly favorable. What was the other kind of concept that we talked about in terms of this reaction? Oh, one of the reasons it's favorable was because we're essentially coupling the phosphorylation of glucose, making this phosphate ester, to the hydrolysis of the phosphoanhydride bond of ATP. So that's one of the things that drives that. If we're just trying to add inorganic phosphate to make that phosphoester, it wouldn't be very favorable. But by coupling it to the hydrolysis of that phosphoanhydride bond, we make it favorable. All right, the next thing I want to talk about, hexokinase, is to introduce this concept of isozymes. So I'm giving you glycolysis, right? And these 10 reactions of glycolysis. And it sounds like there is a standard set of 10 reactions and 10 enzymes that's unwavering, that's similar. But in fact, glycolysis is going to vary a little bit in different cells and different tissues and different organisms and that sort of stuff. What I'm giving you is sort of the general truth, but things might vary. Your experience might vary a little bit depending upon what cell type. And so if we just look at variation in variation in hexokinase in different human cells, we find that there are different enzymes. Most tissues have forms 1 through 3. There's 4 different forms, 1, 2, 3, and 4. And these have low KMs So if it has a low KM, does it tend to bind the substrate tighter or more looser?
Tighter.
Yeah, remember the lower KM, kind of the tighter. It actually means it's the less substrate you actually need to be able to get half the substrate to bind to, half the enzyme to bind the substrate. So that it binds the glucose pretty tightly, okay, but has low substrate specificity. In that it will also binds fructose. So think, an example of a tissue might be a muscle. The muscle has one of these, the major form is one of these forms. And think about the physiological role of muscle is to drive muscle contraction, right? It doesn't have any major regulatory role in terms of maintaining the glucose homeostasis within the body or anything. It just wants to consume glucose, make ATP to be able to contract muscles. That's what it does. And so as a low Km, which basically means it can scavenge the glucose that's coming in from the bloodstream pretty effectively, and it doesn't really care whether it's glucose or fructose. It's just going to bind it to. It's just going to phosphorylate it and so that it can move on and make ATP.
Professor?
Yes.
Forms 1 through 3 of what?
Of hexokinase.
Okay, thank you.
So these are different isozymes, meaning that, we talk about proteins structure, protein sequences. So, and we could say, okay, what's the amino acid sequence of hexokinase? We can come up with the amino acid sequence of hexokinase. These are going to be different sequences with slightly different genes that encode different proteins that are very similar but have slight variations in their amino acid sequence that give them these different properties. So this is the idea of isozymes. Did that make sense?
Yes, thank you.
Okay, so they have these different, and often these different isozymes will have different regulatory properties. I'm not familiar with, there's probably a little bit different regulatory properties of 1, 2, and 3, and I don't know the story behind that. The main story that comes up is liver has form 4, and this has a high Km and is specific for glucose. Sometimes people refer to it as glucokinase because it only phosphorylates glucose. And so then you go back and think, well, why is the liver different? Because the role of liver is to maintain the blood glucose levels, right? So if you eat a bunch of carbohydrates and you have high, your glucose goes up because you've ingested a lot of dietary carbohydrates and now you have high glucose. What it's going to do is it's going to store away that glucose in terms of glycogen so that an hour or two later it can basically restock the blood with that glucose in there, right? It's not trying to generate a ton of ATP to be able to drive muscle contraction or something like this. It's just trying to maintain even amounts of glucose in the blood. So when the blood glucose is really high, this hexokinase reaction will work and it'll enter in metabolism of the liver cell, okay? But when blood glucose is really low, it's not going to work very much because it needs to be going the opposite direction. It needs to be actually releasing glucose back into the cell. So this is how, again, different tissues, different organisms like this will have slight variations upon their glycolysis, or sometimes not so slight variations on the regulatory properties and some of the specific properties of their glycolytic enzymes. What else we like this? We can talk all day just about hexokinase. In terms of well, let's just give ourselves a reminder. Remember our different classes of enzymes. What do we have? Oxidative reductases, hydrolases, like this. Anybody kind of might guess what this might be? Transferase, because we're transferring a phosphate from that molecule to the glucose. It also has an interesting, from a mechanistic standpoint, it was one of the they talk about an induced fit mechanism, where glucose, when it comes in, it binds, it actually then makes the enzyme itself change its structure a little bit, and that change in structure upon binding then orients all the amino acid residues just the right way to facilitate catalysis. All right, we got 10 of them. We got number 1. All right, so the next one, so we mentioned that, okay, well, what do we have to do? Well, one of the things we have to do is split this molecule in half, and I said in order to split this molecule in half, the chemistry is enabled by the carbonyl not being a carbon 1 and carbon number 2. So the next enzyme is going to move the carbonyl from carbon 1 to carbon 2. So we're going to take our glucose 6-phosphate, actually, I'm going to go ahead and write it in the beta form here. Now, we can remind ourselves here that both these molecules are dynamic, right? They're opening and closing, right? Because here's our anomeric carbon. They're open and closing. I happen to write this in the alpha position, all right? But now I'm going to write it in the, no, I'm going to stick it in the alpha position. Sorry about that. I'm going to go ahead. Oh, I said alpha. Yeah. Okay, so now we're going to isomerize this reaction and convert it to fructose 6-phosphate. So this is fructose 6-phosphate. This enzyme is known as phosphoglucoisomerase. You'll see variations of the names of this. Some people will call it phosphohexoisomerase. The delta G naught prime for this, well, I actually don't have the number, but what I'm just going to say is this. Okay, this is thermodynamically reversible. It's pretty close to equilibrium. So what that tells us, so now when this opens up, this is going to be in the, this is going to be a keto group right here, and that's going to facilitate the cleavage of this bond right there. Now some tissues, you know, if you happen to have fructose in your bloodstream, some tissues that are expressing this forms 1,3 with lower specific, like you could take fructose and directly phosphorylate it here to fructose 6-phosphate. The other thing I'll just also kind of mention here in passing, and we'll come back to this, is this ends up being the branch point for glycogen synthesis. So once you make your glucose 6-phosphate, the glucose 6-phosphate can continue the glycolysis, or it can get converted to glycogen down that way. I'm just giving you a preview, and in a week or so we'll get into glycogen synthesis. All right, so now we've added 1 phosphate, but we're going to need 2 phosphates because we're making 2 of those molecules. So the next step is going to be to phosphorylate it again. This time in the 1 position. This enzyme is known as phosphofructokinase. So we take our fructose 6-phosphate, and we're reacting very similar to the hexokinase reaction. We're gonna take a phosphate from ATP, and we're gonna add it now to the number 1 position. So this is now fructose 1,6-bisphosphate.
I have a question.
Yes.
So why is it this phosphate and not that phosphate?
Oh, very good question. Because this will help with the nomenclature. Okay, it's a bisphosphate when the phosphates are in different positions in the molecule. So it's a bisphosphate because the phosphates are here. It's a diphosphate when the phosphates are together. So we take ATP. You know, with our structure of ATP here, and we had P, P, P. So this is a triphosphate because all 3 the phosphates in a row. Actually, if what I should do is draw ADP to get more specifically at the question. So this is ADP. And it's a diphosphate because the 2 phosphates are in a row. Right? This is a bisphosphate because the phosphates are in different places. So, but actually that also brings up another point. Some of these names, they kind of become, they're just like overwhelming. You know, it's like they're all so similar and you know, gluco versus glycero and everything. But if you pay careful attention to the names, they actually make sense. And so if you know things like, okay, well, a bisphosphate means there's a phosphate on 2 different parts of the molecule. You know, the glucose structure versus the fructose structure. When we get to the 3-carbon sugars, it gets particularly seemingly overwhelmingly. But again, if you pay attention to the names carefully, it actually does make sense. Okay, this is another one that is thermodynamically reversible. The delta G naught prime equals minus 15.9 kilojoules per mole. And the delta G, again, because the product is going to be consumed, equals minus 25.9 kilojoules per mole. So this is another one that's highly thermodynamically favored. And from a practical standpoint, it's not reversible. While the phosphofructokinase reaction is easily reversed. Yeah.
Real quick, just because I was curious what it would be. I found a citation for what the delta G naught and delta G would be for the conversion from glucose to fructose. It is cited as 0.4 kcal per mole and 1.7 kilojoules per mole.
Okay, yeah, so under 5. You know, generally if it's like under 5, it's pretty easily reversed. All right, so now we're gonna get to the part, we have the keto group in the right position. We've got the 2 phosphates. Now we can actually split it apart. So this next reaction is fructose 1,6-bisphosphate aldolase. And I'm gonna go ahead and open this up, and I'm gonna show it so we can kind of get a little bit better idea. Okay, so this is the same molecule as this. This is because now in its linear form, fructose 1,6-bisphosphate. And we're going to split it here. And we're going to end up getting so we split this here. This molecule up here is called dihydroxy acetone phosphate. This molecule down here is our glyceraldehyde 3-phosphate. Yes.
Does the dihydroxyacetone phosphate, does the central carbon have another hydrogen?
Oh, thank you.
Question, Professor, when we're doing our map and we're using molecules, can we use the circle P notation for the phosphates?
Yes, yeah, you can do that.
Thanks.
I'll talk about the map in a little bit here. All right, okay, so what else? What else do we have here is going on? Oh, so the delta G naught prime of this reaction is plus 22.8 kilojoules per mole. So is this reaction spontaneous?
No.
No, it's not spontaneous. It doesn't really want to go at all in the direction that we put it here. So it's like, what the heck? How does it work? How does it work if the products get consumed so much that our difference in product concentration and reactant concentration is so high that you can actually drive it forward? So that the delta G is someplace in the order of the delta G is minus 5.9 kilojoules per mole. So this essentially becomes a reversible reaction just because the concentration of the products are consumed at such a high rate that there's a big imbalance in terms of the concentrations of the reactants and the products. All right, so now but now, in this first phase, what we're trying to do is get 2 of these, okay? So the last enzyme in this energy investment phase is going to basically be converting the dihydroxyacetone phosphate to the glyceraldehyde 3-phosphate. And so you just look at those molecules and you basically say, well, we just have to move this keto group from here to here. So it's going to be an isomerization reaction, kind of like our phosphoglucoisomerase, where we converting between an aldose and a hexose, an aldose and a ketose. That's basically same kind of conversion. Let's go over there and do this last enzyme of the preparatory phase, of the energy investment phase. And that is triose phosphate isomerase. And here, now what I'm going to do is just to make it end up with a product with a phosphate on the bottom right here, just like the phosphates on the bottom here. What I'm going to do is I'm going to flip this over. So now we have again, we have our dihydroxy acetone phosphate. Again, the name actually makes sense. Acetone, if you remember the structure of acetone, that would be a methyl group, a keto group, and a methyl group. This is dihydroxyacetone. So there's a hydroxyl group here, and there's a hydroxyl group here. And now this is dihydroxyacetone phosphate, because now we also add a phosphate on there. And this, and again, a very reversible reaction, we end up isomerizing it to glyceraldehyde (3-phosphate). Again, this is a very reversible reaction. The other interesting thing about this enzyme is it has a structure that in which you have a beta barrel on the inside, and then surrounding the beta barrel is in order a bunch of alpha helices in here. And so the active site is right in the middle of the beta barrel, kind of creates, and that structural pattern is actually found in a lot of different enzymes, because you have different amino acids sticking into the middle of that beta barrel to enable the catalysis of different reactions. All right, so that ends, the first 5 reactions, that ends its energy investment phase. And so if we think of our overall reaction of glucose, which is to make 2 pyruvate of glycolysis, we take 2 pyruvates, make some ATP, also have a high energy electron carrier. We haven't really done that. All that we've done, we have 2 reactions that we've used up ATP, the hexokinase and the phosphofructokinase, right? So we're like in an energy hole. We're like this, but they're an investment. We're gonna make our money back. So now we're gonna take glyceraldehyde 3-phosphate, and the first real money maker, I guess. Everybody have this? Oh, let me go. I'm gonna put this one over here. Actually, let's do the same thing on this one. This second phase. The second phase is going to take our 2 glyceraldehyde 3-phosphates and we're going to convert it to 2 pyruvate molecules. And so if we look at this, it's like, okay, well, these no longer has these phosphates in here, right? We've gotten rid of those phosphates. So those phosphates are going to be transferred back to ADP. So we got ADP to ATP. That's going to happen twice. The other thing to notice is we've oxidized this aldehyde to a carboxylic acid. So now that oxidation can be coupled to what's known as substrate level phosphorylation to the generation of ATP. So I'm going to draw that like this, and basically saying that, okay, now we have 2 ADP plus PI goes to 2 ATP, and that's coupled to that oxidation. Again, this is all 2 times per glucose molecule. So we've got a lot going on. In order to go from here to here, we've got to oxidize that carbon. We've got to do some substrate level phosphorylation, and then we got to transfer that phosphate to ADP.
I have a question. Would this be considered step 6?
No, this is the overview. This is a reminder of the overview of the second phase, the energy recovery phase of glycolysis. I just wanted to set it up so that, again, when we think about these next 5 reactions, we can say, okay, what part of this is each reaction doing? And so the first one's going to be a big one. It's going to do this oxidation, convert the aldehyde to the carboxylic acid, or at least the same oxidation state as that carboxylic acid. Let's see. Okay, so the 6th reaction is glyceraldehyde (3-phosphate dehydrogenase. Whenever you have a name of an enzyme known as a dehydrogenase, it's going to catalyze a redox reaction. We're going to be transferring some electrons around. So we have our glyceraldehyde 3-phosphate. And what we're going to do is we're going to oxidize this carbon up here to a carboxylic acid, but it's not like actually the free carboxylic acid is never going to exist. We're actually going to oxidize it and put a phosphate on there, but it's the same oxidation status as if it was a carboxylic acid. So we have NAD plus goes to NADH. And so what we're going to end up with is, I'm going to go ahead and draw the phosphate out in this standpoint, just so I can kind of reiterate that process that so this carbon has been oxidized. So we have this aldehyde right here. You count this has 2 bonds to hydrogen or carbon and 2 bonds to oxygen. This one has 3 bonds to oxygen and another bond like that. So that carbon has been oxidized. Oh, the name of this is 1,3 1,3-bis phosphoglycerate. The glycerate ends up now, it's no longer glyceraldehyde because it's now oxidized, so it's glycerate, but it's phosphorylated, too close, both in the 1 position and the 3 position. 1,3-bis phosphoglycerate. And this becomes our high energy electron carrier. Now I've been referring to NADH for a while now actually as a high energy electron carrier and talked about it in terms of the structure being important for, I don't know if you remember tryptophan. You need a nicotinamide ring in that molecule that can either be made with niacin or from tryptophan, and so that you have a disease if you don't get that. Anyway, I've been talking about this for a little while. Now I want to go ahead and get a little bit of the nitty-gritty here in terms of talking about this. And so what I want to show is, I'm going to show a little bit more of the detail of the aldehyde, and I'm just going to put R here. Now I want to show the structure of the NAD+, the oxidized form of this high energy electron carrier. It's like this, and I'll put R down here. I'll show you the overall structure in a little bit, remind you of the overall structure in a little bit. I'm going to show you that we're going to be bringing in this phosphate here. And what we want to look at is this carbon right here. Because we're basically going to take this hydrogen and these 2 electrons and move it to reduce this carbon right here and make this carbon more reduced. So this is our NAD+, and then this is our NADH. And basically that hydrogen ended up, with those 2 electrons, ended up going over there. We transferred the electrons from this carbon over here and then we end up bringing in the phosphate like so. And then to fully balance it out, take to the fact that we release this, we have an H+. So what I should do in all of these is add the H+ here to be more complete. So now this reduced form of NADH can carry these electrons, and what it'll eventually do is carry those electrons and donate those electrons back to the electron transport chain. It'll become reoxidized to the NAD+. So again, this is the oxidized form here, lacking electrons. This is the reduced form here that contains that extra pair of electrons. And this is reduced relative to this, which is oxidized. So what this ends up, this functional group right here, where we have the phosphate next to the carbonyl here, this is known as an acyl phosphate. This is a particularly high energy bond, even higher than that of a phosphoanhydride. So that can then take us to our next reaction, which is going to be to drive the synthesis of ATP. We can use that acyl phosphate. So one of the characteristics, like when I said, okay, why is ATP such a good energy currency? One of the things I said, well, it's high, that the energy of hydrolysis is high, but not so high that it can't ever be remade. Here's an example of a reaction that's going to actually be made. There's reaction 7, which is phosphoglycerate kinase. It's kind of named for the reverse reaction.